What's up, guys? Welcome to another application session where we show you some of the highest yield content that's on the MCAT and actually how it's tested by AAMC questions and other types of questions. Today it's just going to be me. John's out being a stellar med student, so I'm going to be doing this application session. If you're following along with our program, we're in the Biochem Chapter 4. There's a lot of metabolic pathways in this chapter, and that's kind of what we're going to be going over. The most high yield, what do you need to know about those? Specifically, we're going to be talking about glycolysis, gluconeogenesis, glycogenesis, and glycogenolysis. And we're going to dip our toes a little bit into anaerobic versus aerobic respiration as well. So, let's get into it. Okay, again, if you're in our program, then we're in the BB4 chapter, but if you're not, we're going to be going over glycolysis, gluconeogenesis, and other metabolic pathways. First things first, glycolysis is a hugely important metabolic pathway that y'all need to be familiar with, but there's a lot that goes into glycolysis. I mean, this is just a small diagram of what glycolysis is, but this is a good diagram, in my opinion. Why do I like this? Because it has highlighted the steps where ATP is used. It clearly labels the irreversible steps. You'll notice that those are the same steps in which ATP is used, and then the steps in which ATP and NADH are created. And another irreversible step. So know the net reaction, know how many ATP are used and how many are created, and then how many net ATP there are. Also know the irreversible steps. And I think I have a couple other things labeled. The enzymes associated with the irreversible steps. So I think it's hexokinase on the first one, and then phosphofructokinase, and then pyruvate kinase. So know those and kind of know a little bit about where you're going. You know, when I learned glycolysis, I learned every single step. I made myself write it out specifically. You know, I had an Anki card that said glycolysis, name everything. And I would just kind of sketch out glycolysis. I would say glucose goes to glucose 6-phosphate via hexokinase, and there's an ATP used, and blah, blah, blah. I didn't memorize the structures necessarily, but at the same time, I kind of could, you know, gather that this was glucose 6-phosphate, because I know what glucose is, and I know that there's a phosphate added onto the 6 carbon. So, yeah, I mean, if you don't have glycolysis purpose kind of already in your head, you know, sometimes we can skip over the bigger picture. So know what glycolysis is for. You know, it's the first step in the creation of ATP, or in the creation, in cellular respiration. So it's the first time that glucose is really broken up into anything else. So it turns glucose into pyruvate so that it can be better prepared for pyruvate oxidation, citric acid cycle, and then the electron transport chain and oxidative phosphorylation. Know a little bit about kind of what inhibits glycolysis and what feeds glycolysis. And so that goes back to Le Chatelier's. If ATP is created by glycolysis, it's on the right side of the equation over here. In a net reaction, then having a lot of ATP is going to make it run towards the reactants. That's Le Chatelier's. And so ATP is going to inhibit glycolysis, whereas ADP, which is a reactant that's going to feed glycolysis, is going to make it run in the right direction. And also where it occurs. Glycolysis occurs in the cytoplasm. Once you get past glycolysis in cellular respiration, things start happening in the mitochondria. So keep straight where everything happens in cellular respiration. Glycolysis in the cytoplasm. I'm not going to go too deep into what anaerobic versus aerobic respiration is, but I do want to talk a little bit about how glycolysis plays into it. Coco? I'm not going to go too much into anaerobic versus aerobic respiration. Coco, give me that. I know you're a cat and you like to play, but now is not the time. I'm not going to go too deep into what aerobic versus anaerobic respiration is, but I do want to say glycolysis is in both. So, you know, if you're familiar with the differences in respiration, you know that anaerobic is a lot less energy efficient, like a lot less ATP is created than anaerobic respiration. So the reason why is because anaerobic basically just go through glycolysis where there's 2 net ATP created. And then it has to kind of convert that pyruvate into something else that will replenish the NAD plus so that glycolysis can keep happening. So anaerobic respiration is essentially just glycolysis plus some kind of fermentation. And that's why it doesn't create as much energy. Whereas aerobic goes through all of cellular respiration, you know, glycolysis, like we talked about earlier, pyruvate oxidation, blah, blah, blah. And that's when oxygen is present. So gluconeogenesis, all these G-enesis words can really get mixed up. So break it down. Gluco is glucose. Neo means new, and then genesis means create. You're creating new glucose from other substrates. It could be, you know, fatty acids, amino acids. It could be other things. Whereas glycolysis is where you're breaking glucose down into the other substrates. So I like to think of gluconeogenesis as kind of like the opposite of glycolysis. That sort of is the answer, but gluconeogenesis doesn't necessarily start from pyruvate. It can start from other things. In my opinion, you don't have to know any steps for gluconeogenesis specifically. Just know kind of the body conditions in which gluconeogenesis is favored over glycolysis. And again, stick to your Le Chatelier's. Le Chatelier's is one of the most high yield topics on the MCAT, and you guys should be really comfortable with using it in all scenarios. So conditions include low glucose. So if you have low glucose, then you need to make more. And so gluconeogenesis is going to come in and create that glucose for you. Whereas if you had low glucose, you're not going to be doing glycolysis because you have a low reactant. So the seesaw goes towards the reactants. High ATP and low ADP, again, kind of those opposite conditions of glycolysis. So we have a question here. What body conditions favor gluconeogenesis over glycolysis? I know this is literally just on the slide right before this, but let's think about it. You know, increasing cellular levels of AMP. So I'm thinking if we have AMP, that's kind of like similar to ADP. That's the way I think about it. And so I know that if I have high levels of ADP, that is a reactant in glycolysis. And so it's going to push it towards the products. And so that's going to favor glycolysis. Starvation, that means you have really low glucose probably. So that's going to be favoring gluconeogenesis, where we're making glucose out of things like fats, those long-term energy storages. So probably maybe. High blood sugar. So that's the opposite of starvation, right? You have high blood sugar, that's high reactants on glycolysis. It's going to force it towards the products. And then low cellular levels of pyruvate. So pyruvate's a product of glycolysis. So if you have low pyruvate, it's going to seesaw that way and make glycolysis happen. So starvation's a correct answer here. And I have that highlighted there. Another question, yeast cells can grow on yeast cell. Another question says, Yeast cells can grow under either aerobic or anaerobic conditions at the same concentration of glucose. We're used to grow 2 different yeast colonies. Would the growth rate be faster under aerobic or anaerobic conditions? So if you were going to simplify this down, you could basically just have the last sentence. Would the growth rate be faster under aerobic or anaerobic conditions? Which conditions are going to help whatever process we need to grow? So let's kind of see the answer choices to see if that's enough of a simplification. A says anaerobic because the final product alcohol would contain more energy than the final product of respiration. Well, remember that those final products are not necessarily what we care about. We only care about the energy that's created kind of in the interim. So no, anaerobic because the cells would not have to produce the enzymes needed for the citric acid cycle. I don't think that this is necessarily something that makes aerobic respiration less energy efficient, like having to produce those specific enzymes. Like we make so much more ATP with aerobic respiration. So I don't think that that's a valid answer. C says aerobic because a much greater amount of ATP will be produced to provide energy for reproduction. I like that one. It makes sense. It's simple, straightforward, and it has all of the aspects I'm looking for in the right answer. D says aerobic because the CO2 produced in fermentation would be toxic to the culture. I don't know. I don't think CO2 is toxic to yeast. We would never have bread if that were the case. So I like C better personally. Okay, let's take glycogenesis and glycogenolysis. Those words are so similar. Let's not get them confused. Break them down if you're ever getting confused about which one you're looking at. So glycogen, we have glycogen sort of in both of these words. So we're talking about glycogen, the long-term storage for carbs. Genesis means create. So in glycogenesis, we're creating glycogen, whereas in glycogenolysis, lyse means to break down. So in glycogenolysis, we are breaking down glycogen. They are the opposite of each other. So what body conditions are going to favor each one of these? If we have lots of glucose, plenty of glucose, then we need to store that somehow. And so that's going to favor glycogenesis, where we're creating that glycogen, storing it in our liver and in our muscle cells. And that's sort of a medium term, not necessarily long term, because long term would be like fats, but it's sort of a medium term carb storage, things that can be used within like 24 hours. I don't know the exact hour amount, but it's something like that. Whereas when glucose stores are low, we're going to be using that stored glycogen that we made earlier, and we're going to be breaking it down via glycogenolysis. This question says, at birth, the serum glucose concentration of a normal infant. So I put a little bit of a passage up here on the top, and then I put the question down here on the bottom left and the answers right here on the bottom right, in case you are not familiar with the way that we kind of run these application sessions. So the passage says, at birth, the serum glucose concentration of a normal infant, approximately 4.5 millimolar. So the passage says, at birth, the serum glucose concentration of a normal infant, approximately 4.5 millimolar, is only enough to support normal metabolism for 70 minutes. After the depletion of the blood glucose pool, the initial source for energy replacement is from hepatic glycogen. Hepatic means liver, so that's basic science, which is sufficient to support basic metabolism for the first 12 hours. Mobilization of these stores is dependent on a rapid increase in the level of glucagon and activation of its 66 kilodalton G protein coupled receptors in the liver. Because infants normally do not receive an adequate supply of glucose from feeding for the first few days of life, hepatic glycogen stores are soon exhausted and the glucose pool must be replenished from non-carbohydrate sources, including fatty acids and glucogenic amino acids. Okay, so we have a little bit about embryonic so we have a little bit about fetal or infantile metabolism. So the question says, based on the passage, which metabolic pathways are upregulated during the transition from intrauterine to extrauterine environment? Oops. Okay, so to simplify this question, I need to know what is the transition from intrauterine to extrauterine environment. So let's go back a little bit. So it looks like it says the first thing that we do is basically glycolysis. You know, we use the glucose that's in our blood. The serum glucose concentration, that's what we're using for glycolysis. But after the depletion of that, then we use hepatic glycogen. So what is, when we're using glycogen for energy storage, what are we doing? We have to break it down into glucose first. So that is glycogenolysis. So first we have a little bit, very short period of glycolysis, followed by glycogenolysis. Do we have anything else next? I seem to remember there was something else, blah, 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 something about a G protein. And then the glucose pool must be replenished from non-carbohydrate sources, including fatty acids and glucogenic amino acids. So that is a huge indicator that we're going to be creating glucose from something that is not glucose. And so that would be gluconeogenesis. Gluconeo, I'll just write that out. So let's see if any of our answer choices say this. Glycogen, A says glycogenolysis. A says glycogenesis followed by glycogenolysis. So we're not ever creating glycogen in this. We're only searching for energy storage or ways to use the energy. A says glycogenesis followed by glycogenolysis. So we're not ever making glycogen in this passage. You know, we need energy now. We don't have energy to create glycogen from. We need energy now. So we're not doing glycogenesis first in any way. Or B says glycogenolysis followed by gluconeogenesis. Okay, well, I see that that's kind of what I have right here in the second half. It doesn't mention that little 70 minutes of glycolysis. Not sure why, but it doesn't. But I know that B is the order in which my metabolic pathways are lining up in this passage. C says gluconeogenesis followed by glycogenolysis. So that's kind of the flip, right? But we're not actually creating glucose from the non-carbohydrate sources first. We're actually using our glycogen first. And so B is a better answer. And then D says gluconeogenesis followed by glycogenesis. So again, we don't do glycogenesis in this passage. So B is the right answer here. This question says, activation of which enzyme would support the metabolism of newborn infants during the first 12 hours? So again, this is talking about glycogenolysis. So we're breaking down our glycogen. So basically this is asking what enzyme is most important in glycogenolysis. So, you know, you kind of have to know a little bit. These are all around surrounding glycogen, so that doesn't give us any hints. So you have to kind of know a little bit about what these enzymes are. If you're using the content guide that we've given you, there was a great quote about the correct answer here. But what you need to know is that glycogen phosphate. You're so cute, but I'm doing something right now. Glycogen phosphorylase is going to be the enzyme that kind of puts inorganic phosphate onto the glycogen to kind of break it off from its glycogen chain and break it into a glucose monomer. I think I have the actual quote down here. I don't know, it's not showing up on here, but it says glycogen phosphorylate uses inorganic phosphate in order to cleave alpha 1,4 glycosidic bonds to produce glucose 1-phosphate. So that's the purpose of glycogen phosphorylase. Phosphatase, y'all will have to fact check me, but I'm pretty sure phosphatases take phosphates off of groups. And then kinases transfer, but in this case, we're using an inorganic phosphate, just one that's kind of floating around, and we're putting it on glycogen in order to kind of break off a glucose monomer. So in that case, glycogen phosphorylate would be the right answer. That'll be it for this application session. Thank you all for joining in. If you have any questions about metabolic pathways that you need to know, drop them in the comments below. Otherwise, like this video if you like free MCAT practice. All right, bye.